So this is almost the part that everyone's been waiting for, and this is the discussion of the tertiary structure of a protein. The tertiary structure, which I've already kind of led into a little bit, but we're going to get into a little bit deeper here. Tertiary structure refers to the overall spatial arrangement of atoms in a protein. This is stabilized by numerous weak interactions between amino acid side chains, largely hydrophobic and polar interactions, which are actually going to do it, but they also can be stabilized by disulfide bonds. And that might seem like disulfide bonds. What? Where did that come from? That seems like a little bit of a curveball. But remember, cysteine is a residue that has a sulfhydryl group. Now, that sulfhydryl group with another sulfhydryl group can form what's known as a cysteine. So that is basically a way that you can form a covalent bond between two amino acid side chains. So interacting amino acids are not necessarily next to one another in that primary sequence. So this could be cysteine number 212, 212, and this could be cysteine 396. It doesn't even matter. They could be very far from one another in the primary sequence because it's all about the tertiary structure in which it's all about the three-dimensional shape that a protein takes on. Now, proteins are going to take on two major classes. They're going to be fibrous and globular. Those are our two major ways. So globular proteins are going to be water or lipid soluble. So that means that our fibrous proteins generally are not going to be water and lipid soluble. Now, a question for that is why not? Well, that has to do with kind of the structure of those proteins. This is an example of a fibrous protein that has what we've got here is a total of 3 helices that are basically wrapped around one another. So this helix is very tightly wound. It's got 3 amino acid residues per turn. This is glycine, proline, and a derivative of proline. These are 3 helices that are wrapped around one another. Now, one thing that you should know about this is these residues are not very water soluble, hence why our fibrous proteins are not water soluble. Now, fibrous proteins from structure to function, structure of them, an alpha helix is cross-linked by disulfide bonds. These are very tough and rigid, and they are found commonly in keratin of hair, feathers, and nails. The beta conformation of a fibrous protein is going to be soft and a little bit more flexible, and that's going to be found in something like a silk fibroin or a collagen triple helix. That's so that's found in collagen tendons. These are very strong proteins that are not going to stretch very well at all. So this would be a cross section of a single strand of hair, and it's going to be kind of broken down. Let's look in the front. This is an alpha helix, and this is a 2-chained coil. So 2 of these alpha helices wrapped around one another, which wrap around one another to build up this protofilament, which wrap around one another to form up this protofibril, and they're going to wrap around one another to form an intermediate filament to ultimately come together to make an entire hair strand. So they're very strong and tightly wound. So and if you think about putting hair into water, well, that's not going to go into that solution very well. Instead, it's just going to float at the top. So here's a little bit more of an investigation or a dissection of keratin structure. So alpha keratin in the hair, it consists of that 2 alpha helical or 2 helical structures that have wrapped around one another. That kind of lowest level is your 2-chain coiled coil. Those 2-chain coiled coils, those are going to basically wrap together and organize to form a protofilament. And those protofilaments will be kind of the base units that build up to form these protofibrils. If you look, the ends of these protofilaments, what you're going to see is those ends are right here and right here and right here, respectively. So the protofilament is multiple 2-chain coiled coils. The protofibril is multiple protofilaments that have kind of all assembled together to make this larger structured molecule. Now keep in mind, just for the sake of scope and scale, what we're looking at here with the protofilament is something that's approximately 20 to 30 angstroms in terms of its height there. Okay. So curly hair is actually something that gives a little bit of a connection between the single amino acid level and kind of the higher order like visual level, so to the naked eye. So the presence of cysteine residues and the formation of disulfide bonds between cysteine residues on adjacent proteins and adjacent alpha-keratin protein strands is actually what causes permanent waving of hair. And so curly hair is the result of a higher presence of cysteine residues. So in order to disrupt that, like if you use a hair straightener or something, I think that's what they're called. I actually don't use them. Despite having curly hair, I've never, yeah. So anyway, you would reduce those disulfide bonds, so you have these cysteines with the sulfhydryl groups, and then ultimately you could rearrange them, you could curl them to oxidize them. So the treatment to, I guess a perm, I think, I'm not really a hair person. So basically the curling of hair is the result of disulfide bonds being formed.